So our next talk is another one that really kind of harkens back to days of JGI past. One of our very earliest metagenomic projects at the JGI was studying the symbionts of this gutless worm that Nicole is going to talk about next. And again, it was a case where we got some fascinating insights, but really couldn't dig as deep as we wanted to with the technology available then. And in the last few years, we've revisited this subject and really gained a whole new depth of knowledge about these symbionts that Nicole Dubilier from the Max Planck Institute is going to be telling us about today. Thanks.
Thank you for the introduction, Susanna. I'm going to be talking about chemosynthetic symbioses, and they were first discovered in 1977, and they completely revolutionized our understanding of the energy sources that can fuel large communities of life on Earth. Because previous to their discovery, it was assumed that light energy, energy from the sun, was essential for fueling large communities, and therefore their discovery was one of the most remarkable discoveries made in recent time about what kind of communities can thrive on Earth. These thrive in the deep sea, and they were discovered in 3,000 meters water depth, thanks to submersibles that were able to go down to these depths. And we now know that they can support tremendous biomass. It's at least as large as tropical rainforests. Is this going on and off sometimes, or is it okay? It's okay? Okay. As coral reefs and kelp forests. And the reason that the reason for this huge biomass is due to symbiotic associations between bacteria and marine invertebrates. And how does chemosynthesis work? The bacterial symbionts provides their host with a source of nutrition via chemosynthesis, and what they're doing is they're fixing CO2 into biomass using reduced inorganic compounds such as hydrogen sulfide as energy sources. So if you compare it to photosynthesis, what you have in photosynthesis is the sun that's providing the light energy. The chloroplasts, which are essentially reduced symbionts, are fixing the CO2 into biomass and feeding the plant. And when you look at chemosynthesis, then you've got a similar process, except it's something that we call dark energy, not dark matter, but dark energy, chemical energy that is coming at hydrothermal vents from the interior of the Earth, reduced sulfide that is used as an energy source. The symbionts fix CO2 into biomass, and then the symbionts are either digested by the host or the host milks the symbionts to gain nutrition then from the symbionts. So we had to actually go 3,000 meters down to the bottom of the ocean sea floor to discover these types of symbioses, but the irony is that they were occurring in our own backyards and shallow water sediments for millions of years, and that we weren't aware of them as biologists. We now know that they're widespread, occur in shallow waters, in seagrass beds, mangrove swamps, coral reef sediments, and even in sewage outfalls where there are many reduced compounds that can be used. And today in my talk, I'm going to very briefly talk about two of the model systems we work on. We do work on animals that have at least a partial gut. These are mussels in the deep sea, and then I'll spend a little bit more time talking about the gutless worms that we've been working on. Okay, so just very briefly, a little bit more about chemosynthetic symbioses. Numerous bacterial lineages have been able to establish these symbioses in convergent evolution. So there's a strong selective advantage for the bacteria to establish these associations. They occur in gamma proteobacteria, alpha proteobacteria, epsilon, spirochetes, and even within these divisions, there are multiple lineages that have been able to establish these symbioses. And at least 9 animal groups and also several protist groups have been able to establish these symbioses in convergent evolution. And these are just some of the hosts that have chemosynthetic symbionts, nematodes with ectosymbionts and ciliates and platyhelminths, and then many different kinds of shrimp and clams and mussels and our worms here. Okay, so in the first part, talking about the mussels, I'd like to talk about what I call from the environment to omics. And so I was trained in environmental microbiology, and what we do is we look at an environment and we can usually have a good idea about the energy sources that are available in that environment, and then we look at how the microbes are using that. And that's our classical approach in environmental microbiology. And so this is the approach that we used to look at the energy sources that are being used by these deep sea mussels from hydrothermal vents. And they produce, as I said, huge biomass. It's 2 to 5 times as much as in their shallow water relatives in Mytilus, in Mytilus, Edelus, because of their chemosynthetic symbionts. And this is what it looks like. In these mussels, they live in a dual symbiosis with 2 types of intracellular symbionts. And here you see a fluorescent in situ hybridization image where we've used probes that are specific to the methane oxidizing symbionts that they have and the sulfur oxidizing symbionts that they have. And these 2 symbionts use the corresponding energy source, so methane as an electron donor and the sulfur oxidizers reduce sulfide compounds. And what you see here, by the way, are then the host nuclei stained with DAPI and a general eukaryotic probe that is highlighting the areas in the gill where there are no symbionts. Okay. So we hypothesized that hydrogen could be an energy source that could be used by the symbionts based on the following. And I should say that previous to this, to our discoveries, only 2 reduced compounds were known to fuel these symbioses, reduced sulfur compounds and methane. And that was always discussed at meetings on chemosynthetic symbioses that we said, well, we don't really understand why that is because there are many other energy sources, reduced compounds that are available at hydrothermal vents, and there are many free-living bacteria that are able to use them. And particularly hydrogen is abundant at many vents, and it's energetically incredibly favorable. Most bacteria suck up hydrogen quickly if it's around. What we did was thermodynamic modeling, where we simply looked at the temperature range where the mussels occur and modeled what the energy yield from hydrogen would be in comparison to methane and to sulfide. And you can see that if a bacterium is able to use hydrogen, it can gain 7 times more energy from hydrogen than from methane, and 18 times more energy from hydrogen sulfide. And so we were lucky enough to get funding to go to a hydrothermal vent on the mid-Atlantic ridge where hydrogen concentrations are remarkably high. And what we were able to show is that indeed hydrogen is an energy source for these symbioses. And I'm going to actually go through this relatively quickly because we published it a while ago and because I want to move on to the gutless oligochaetes because we have some new stuff there. But what we are able to do with incubations on board to show that hydrogen sulfide is, that hydrogen is indeed used or is taken up by the gill tissues, that they fix CO2 in the presence of hydrogen. We were able to amplify the key genes that are used for hydrogen uptake, and then we were able to show using in situ hybridization methods that it's the sulfur oxidizer that is able to use the hydrogen, not the methane oxidizer, the sulfur oxidizer. And then using deep sea in situ mass spectrometry in collaboration with Pete Gerges from Harvard, we were able to show that the mussel, over the mussel beds, we see a depletion in hydrogen, indicating that hydrogen is being used. And after having all of this data, we finally got our metagenomic information from Genoscope in France. The irony of this, and I'm going to go ahead and say this, is that we submitted a proposal to the community sequencing project at the time that was rejected, which is why we went to Genoscope. And they were able to then give us or confirm with the metagenome that it is indeed the sulfur oxidizer that has the genes that are needed for CO2 fixation and for using hydrogen as an energy source. Okay. And so, as so often when you discover something like this, what you see is that it's not only in your organism and that many other symbionts and symbioses are able to do that. And so we now know that these giant tube worms that are very abundant also use hydrogen as an energy source, and that shrimp can also use it. And furthermore, now in a very nice metagenomic analysis done by Greg Dick together with transcriptomic analysis, he was able to show that the free-living bacteria that are there in the vent environment are also actively using hydrogen as an energy source. Okay, so now I'd like to do it the other way around, which is how omics can tell you something about the environment that you don't know previously. And really, as an environmental microbiologist, that can be quite an experience, and I'll try to explain what we were able to see. Okay, so now I'm talking about the gutless oligochaetes that occur in shallow waters, in mangrove swamps. This is what they look like. They're fairly small, about 0.5 millimeter in length and about 2 to 3 centimeters long. And they occur in the pore waters of marine sediments, so they're interstitial. They're moving up and down between the oxygenated and reduced sediment layers. And they've completely reduced their digestive system because of the symbiosis. They have no mouth, no gut, no anus. And also particularly unusual is that they've reduced their excretory systems, their nephridia. So their bacterial symbionts are not only feeding them, but they're also taking care of the waste products. And we've described, or there are more than 100 species that have been described from around the world. They're monophyletic, which means that the symbiosis evolved once and has now diversified. So it's a great group to also look at the evolution of these symbioses. And they live in symbiosis with 5 to 6 co-occurring symbionts. Each species has its own specific assemblage of symbionts, but they differ between species. And they're ideal low-diversity microbial ecosystems. So you heard 2 talks ago bioreactors and how you can look at them. And what we say is that each little worm is a bioreactor. And we can take that little worm and specifically analyze the microbial community that is in it using many of the techniques now that are available for looking at that small quantity of a microbial ecosystem. Okay, so I'm going to focus in now on Olavius aigavensis that occurs. It was actually first discovered off the coast of the Algarve, but there we've never found it again. And our main sampling site here is on Elba, Italy, in the sediments there. This is the bay that we sample them in, and what is important here to look at is these dark patches are seagrass, and that's going to play a role in what I'm going to be talking about. This is Tanja Voige sampling them back in, way back in, what was it, 2004. I think this is the only field trip that Eddie allowed Tanja to do. And I think if he had known how serious we are about our work hard, play hard approach while sampling, I'm not sure that he would have really let her go. Okay, so the morphology of the symbiosis is that the bacteria occur in a thick layer just below the cuticle. What you see again here are probes that are specific to the different types of bacteria that occur there. So that the epidermal layer that is normally just underneath the cuticle has been pushed down, and there's a huge layer of these bacteria around here, and that they're not inside the cells, they're not intracellular, they're just below the cuticle, so they're exposed to what's in the pore waters and have easy access to it. And so when I first started working on these worms in 2001, what I found was that they had the classical sulfur oxidizing symbiont that is well known from chemosynthetic symbioses, the sulfur oxidizer that uses reduced sulfur compounds. But one of the things that I spent probably 2 years looking for were reduced sulfide concentrations in the environment, and we could never measure them. They were in the range of what you find in open seawater, so nothing that we could possibly imagine could be driving this type of symbiosis. And then it took a couple of years until I was able to show that they have a sulfate reducer. So the bacteria that produce reduced sulfide compounds in the environment was taken up in evolutionary time by the host and had now evolved into an endosymbiont that was producing the reduced sulfide that is needed by the primary symbiont. And so we have a sulfur cycling going on with the reduced sulfide compounds being produced by the sulfate reducer that are feeding the sulfur oxidizer. And it then took another couple of years for us to show that it wasn't 2 symbionts that co-occurred, but that there was a second sulfur oxidizer that co-occurred, a second sulfate reducer, and a spirochete. Okay. So I was very excited in 2004 or 5 when my proposal to sequence these symbioses was accepted. And this is not a mistake with MB. This is Sanger sequencing times. And it was also a time when we needed at least 200 worms, a pooled sample, to create enough DNA for a single run with phosmid libraries and creating this. So it was really, it's been incredible for me just within this short time period how far we've come. But I still think there's a lot to be said for Sanger sequencing, but I won't go into that. So at the time, what was quite novel was using binning approaches in metagenomics to tease apart then the genome fragments that you have, and we used methods that by now are quite standard. The one thing that I would like to point out is that we only worked in our analyses with the sequences that we could bin, which was about 20 MB. And about 9 MB of them were not binnable. So that entire wealth of genome information was something that we didn't analyze at the time. And the metagenomic coverage of the symbiont genomes was about 30 to 90%, very variable. And we had a ton of open questions from the metagenome. Essentially, as you all know, genomes really just give you hypotheses to work on. But one of the things that we were very interested in is why is there redundancy in symbiont function? Why do they have 2 sulfur oxidizers, and why do they have 2 sulfate reducers? And the other question that we could not answer based on the genome was, what are the environmental energy sources that are driving the symbiosis? We knew that there was no sulfide in the habitat, but then what's feeding the sulfate reducer that would feed the sulfur oxidizer? And based on the genome, hydrogen could be an energy source that they could use, but hydrogen has never been shown to be present in marine sediments in any kind of high concentrations. So then we postulated also that the sulfate reducers could be using organic carbon, but in these poor waters in the Mediterranean, they're completely oligotrophic. And when we first brought our samples to our metabolomics collaborator to measure, he said, so I think you've given me millicu. There's just nothing in there that is really available. And so that's where we started using metaproteomic approaches to work on these. And Manuel Kleiner, who you see down here and is here at the meeting, is the person that has really been driving this metaproteomic work and doing a brilliant job on it. Cecilia Ventrup from my group, who also worked on these, and Christian Lott at the Hydro Station, who's been giving us great possibilities for collecting these worms that we actually hand pick out of the sediment and collect and then look at. Okay, so very briefly, one of the nice things that Manuel was able to develop was something that he called proteomics-based binning. So what he was able to do is identify about 1,600 proteins based on those sequences that we had already binned. But he was also able to identify another 1,000 additional proteins by looking at those proteins that were abundant in certain symbiont fractions, then going back to the unbinned part of the genome and looking for them there, and then carefully doing proteomic genomic comparisons to show that these proteins were indeed coming from the symbiont that he believed they were coming from. And so we were able to confirm a lot of the predictions that we made based on the genome, but we were also able to make a number of novel discoveries. And what I'd like to very briefly just generally talk about is the power of proteogenomics. So anybody who's done an annotation of a bacterial genome knows how overwhelming it can be, because they can be heterotrophs, they can be autotrophs, they can use all sorts of energy sources, they synthesize their own lipids, their amino acids, and what you're really looking at is a huge forest, and it's very, very hard to tease it apart. And so what you get when you're looking at proteomics is that you start to see pathways. You're looking at proteins that are abundant, and they're popping up, and they're literally highlighted like this. And what's also wonderful about the proteomics is, for example, here in our symbioses, we saw the Calvin cycle genes being highly expressed as something you would expect in an autotrophic organism. But what Manuel saw, which was very interesting, was that although the Calvin cycle was highly expressed, there were 3 key enzymes of the Calvin cycle that were missing and that he could not find in the proteome, and also went back to the genome and could not find. And instead, what he did see was that there is a trifunctional, or that is hypothesized to be trifunctional, so that it would substitute for all these 3 enzymes, a pyrophosphate pyrophosphatase-dependent 6-phosphofructokinase that is replacing these 3 enzymes. And the postulation that Manuel has is that it could save up to 30% energy for organisms that are able to use it. And using then comparative genomics, Manuel has shown that this is widespread, this trifunctional enzyme in many autotrophic bacteria. And so that really was not visible in the genome. When you do the proteome and when you compare it, that's when these things start to really pop up. And so the other story that I would like to talk about is that we discovered that carbon monoxide appears to be a key energy source in this symbiosis. The gamma-3 symbiont, so the second sulfur oxidizer, expresses a carbon monoxide dehydrogenase that it can use under aerobic conditions. And the 2 sulfate reducers express a carbon monoxide dehydrogenase that they can use under anaerobic conditions. And we were really quite excited about this because carbon monoxide has not shown previously to be an energy source in animal microbe symbioses. And obviously, when you think about the fact that carbon monoxide can be a bit toxic to aerobic life, it's not something that you would normally think about. And so what we then asked is, now that we see it in the proteome and in the genome, is it present in the habitat and in the environment? And what you see here is a drawing of where the worms occur, and they're very abundant just above these dead seagrass roots, the rhizome roots that are really, really abundant in the sediment. They do move up and down, but you find them mostly just above the rhizomes. And then using pore water samplers, we sampled the pore waters there. And indeed, what we were able to show is that the carbon monoxide concentrations above the rhizomes, where the worms are very abundant, are quite high and considerably higher than those that are normally known to exist in coastal sediments, 15 nanomolars, and we often measured up to about 60 nanomolars in carbon monoxide. And so what I like to say is that for me, as an environmental microbiologist, it's really ironic that it was actually the proteome that was able to show us that there are energy sources out there in the environment that we had no idea about and that are playing an important role in driving the community. And we're now looking at the free-living microbial community because we're convinced that this carbon monoxide is actually driving the microbial community in these seagrass sediments. Okay, so the symbiosis we now know uses multiple energy sources, carbon monoxide, hydrogen, reduced sulfur compounds, with carbon monoxide being used by 3 of the symbionts. Hydrogen is definitely being used by at least 2 of the symbionts. I don't have time to talk about the fact that we have incredibly high hydrogen concentrations in these sediments. We think it's a microbial process that I won't explain here. So in terms of understanding the symbiont diversity in the symbiosis, why they have so many symbionts, multiple symbionts enable the association to exploit numerous energy sources. And what appeared to be functional redundancy based on the genome was not when we actually looked at the proteome and looked at the metabolic pathways that are being used by the symbionts. We published this recently in PNAS. I think, how much time do I now have?
We've got 7 more minutes.
7 more minutes. Okay, great. So if I talk even faster, I'll be able to tell you a little bit about our first results now with the community sequencing project that was accepted in 2012, where we're starting to look at the symbioses in a little bit more depth in the gutless worms. These are the many different sites, red spots, where they've been found around the world. These are the sites where we've already collected them. It's really a shame that Tanya hasn't been able to go out more often and help us collect them. Or maybe we should just convince Eddie to come out there with us and collect them, because some of them are really quite beautiful. One of the problems that we have that might be interesting for those that are working in very distant communities or places where there's not a freezer or dry ice, it's something that we always have a problem with, is that RNA later works really, really well for us for then doing, I see lots of people nodding and have already discovered this, but we were actually quite pleased when we were no longer dependent on getting liquid nitrogen out to these places. We've been using all sorts of approaches now to try to complete their genomes, fact sorting, sorting then of the cells, but individual and collecting cells and density centrifugations, but also individual worms and DNA extraction. We've tried, I think, probably every method at the moment out there, including microfluidic single-cell sorting in collaboration with Paul Blaney and Steve Quake and all sorts of different methods that you can see up here. And essentially, at the moment, what it looks like is that for every symbiont, a different method seems to be working well. So it's not over the board, easy to say what method is working well. So as an example here's some of the first data that you can see for the spirochete genome that I didn't talk about. And what you can see here, and which kind of makes sense, is that if you're working with a single cell, then you're going to have the contig length and cumulative length is going to be relatively low. Here, metagenomes work quite well. For other symbionts, it's looking like the FACS sorting of up to about 1 million cells seems to be working better, and for other metagenomes. I personally am very excited about what we can do with looking at the metagenome of a single individual. One of the things that's beautiful when we do the binning is that the mitochondrial genome just literally falls out into a nice closed contig that we can look at. And so in terms of doing phylogenomics, this is a really easy and cheap way to look at many, many different kinds of organisms, is simply by looking at the entire metagenome of the worms. Another thing that is advantageous about using metagenomics is that we can look at the abundance of the symbionts in individual worms. And anybody who's tried to develop primers and do qPCR for trying to look at the abundance of changes in a community knows how time-consuming this is. And with the metagenomes, what we're seeing is, for example, here a delta-3 symbiont that we thought was just not important and that we thought was a contaminant, and that apparently in some worms really does appear to be abundant and might be playing a role. So additional symbionts that you previously were not aware of. And then here, the high coverage that we're seeing, for example, for the carbon monoxide. This is what we had from Tanya's paper in 2006 in the Nature Metagenome. We had the genes that were there for the oxidation of carbon monoxide, but they were at the end of 2 contigs and they weren't complete. And now with resequencing, what we're seeing is a nice, clean carbon monoxide operon that is used for carbon monoxide oxidation. Okay, so in summary, I hope I've been able to convince you that these types of symbioses have enabled animals to thrive in environments that they could not otherwise live in, and that they really contribute to ecosystem functioning and fueling diversification of life on Earth. I have quite a few PhD and postdoc positions available in my lab, so if you're interested, please contact me, and I'll finish with the Homo sapiens symbiosis. This is my group at a retreat we just did in January, and I think I've named quite a few of the people that have played a role in contributing to these results. Thank you.
All right, well, that was a great talk. Do we have any questions? I think we have time for maybe 1 or 2. Wow. Well, I guess everyone wants to get to lunch.